Ladies and gentlemen, please welcome SC09 Biocomputing Chair, Peg Falter.
Good morning, and welcome to the second day of the SC09 technical program. Today's technical thrust area is biocomputing. Beginning with this morning's plenary speaker, you will be listening to invited talks, papers, panels, BOFs, and elements of the exhibitors forum that will describe how advanced computing has enabled societal impacts in the biological sciences. The first half of this morning's session, you will hear from Lee Hood, a visionary in the field of genomics, genetics, and biotechnology. And immediately following his talk, we are going to be having the first ever presentation of the Kennedy Award in honor of the late Ken Kennedy. When I first joined the Human Genome Program over a decade ago, Lee Hood was already considered as one of the cornerstones in the field of genomics. He received his MD from Johns Hopkins and his PhD from California Institute of Technology. I'm sure everyone here is aware of the Moore's Law style growth in DNA sequencing. What you may not know is that it was Lee and his colleagues from Caltech that was responsible for the first DNA sequencer in addition to the DNA synthesizer. That has resulted in not only the completion of the Human Genome Program, but the completion of thousands of genomes sequenced and the many discoveries from that information. Lee is also a pioneer in the field of systems biology. Systems biology is really adapting or considering and taking into account all aspects of the systems within a living organism. This is a marked change from the previous type of research in the area, which was very specific gene-centric. Lee has co-founded over 20 biotech startup companies, including Amgen, BE Biosystems, the Institute for Systems Biology, which he remains president, and his latest startup, Integrated Diagnostics, or INDI. INDI was just announced last month with a $30 million startup aimed at early detection of cancer and diseases such as Alzheimer. And if we can get a $30 million startup in this economy, that's a pretty good accomplishment. I think we'll be hearing a lot about this latest startup and the work they're doing there during his talk, which is entitled Systems Medicine, Transformational Technologies, and the Emergence of Predictive, Personalized, Preventative, and Participatory Medicine. Please join me in welcoming Lee Hood.
Well, thank you very much. I'd like to discuss with you a revolution that's coming in medicine, and I think it will largely be done over the next 10 years or so. This new medicine that we call P4 medicine, together with the emerging technologies in which it is embedded, will fundamentally change medicine from a reactive discipline to a proactive discipline. And of course, the essential change is medicine will become an informational science. We'll be talking in 10 years about billions of data points on each single patient that will allow us to search health space and disease space in ways that are today utterly inconceivable. To do that, I'd like to talk a little bit about how I view systems biology and systems medicine. I'll give you one example of how we've taken a systems approach to disease and the transformational insights that that's provided us. And then we'll talk about three of these emerging absolutely transformational technologies before we end up talking about P4 medicine. It's clear what has led to this thing we call P4 medicine are the convergence of several different technologies and strategies for doing biology. The Human Genome Project, for the first time, gave us a complete parts list of all genes and, by inference, all proteins. And, of course, this was necessary for any fundamental holistic systems kind of approach to understanding biological function. The changes that have occurred in computing, particularly with regard to their applications to biology, the changes that are occurring now in measurement and visualization technologies, and finally this whole discipline of systems biology have led us to P4 medicine. And the essential underlying framework of P4 medicine is this idea of biology as an informational science. And let me talk about two fundamental types of biological information. Of course, the first is the digital information that's embedded in your genome, and the second is the environmental information that impinges upon that digital information. The two together in an integrative manner create development, create physiologic responses, ultimately create the phenotype that are each of us as living organisms. And of course, it's the integration of these two types of information that will give us insight into disease as well. Information is mediated by two types of biological structures. The first are biological networks that capture information and transmit it, process it, integrate it, and then finally pass it off to simple and complex molecular machines where that information is executed. And in systems biology, understanding the dynamics of these networks, how information is managed, and as well how it is passed off in an effective manner to the molecular machines is really the essence of this new discipline. What I think underlies the real heart of systems biology is a cross-disciplinary environment where you can have biology and the frontiers of biology be the driver for the type of technologies you develop, and correspondingly for the type of computational and mathematical tools you need for dealing with biological information. And this is kind of the holy trinity of systems biology, but it is, I stress, driven by biology. What you need in this cross-disciplinary environment are all the types of scientists, physicists, engineers, computer scientists, chemists, physicians, and the like, because the idea is to work together in teams to take on hard problems where it's not just solving the biology, it's developing the technology and computational tools you need to move toward this information-based, in this case, biology as well as medicine. I would say there are 8 fundamental features to systems biology. The first is we have to be able to do global measurements. We want to look at all genes, all proteins, all whatever informational structures are present in a living organism, because we don't know enough about any complex system to selectively say we'll study only a subset of the information. The second important idea is that there are many different types of biological information, from DNA to RNA to proteins, networks and interactions, up to cells and organs and individuals. And at each of these levels, the core of digital information manifests and is modified by input from the environmental information. And to understand this integrative, multi-scalar, successive hit of digital information with the environmental information, we have to carry out a process of integration that delineates clearly these two types of contributions. We also have to remember that these networks are really dynamic, and we have to develop new tools for being able to measure the dynamic nature of these networks. They change their architecture as well as the molecular constitution of their key nodal points. Systems biology is fundamentally hypothesis-driven. You take all the information about a system, you create a model, you test that model with hypotheses, and you carry out systems perturbations to test these ideas. You reintegrate that back into the model, and the idea is you do this in a successive manner, iterative manner, until you bring theory and reality into apposition with one another. It's really important to understand that ultimately what you want to do with models are make models that are not only predictive, but in the case of medicine, are actionable. And of course, the models can be descriptive, they can be graphical, they can be mathematical, depending on the amount of data that you have and so forth. Discovery science is a key part of systems biology, taking a biological object and defining all its elements. The Genome Project was the classic example of discovery science, not driven by hypotheses, just driven by what is the reality of 24 digital strings of a 4-letter alphabet of DNA bases and so forth. It's absolutely key that the measurements that we make at these various multiscalar levels be optimized and parallelized so that we can get high throughput information capture. And of course, what is really important in any large data gathering process is dealing with the signal-to-noise issues. And in biology, the noise issues are absolutely overwhelming and frankly can only be dealt with by really clever biology, and I'll show you an example of that. So at ISB, for the 10 years or so of our existence, we've used the biological problems to drive the development of pipelines for data capture, to be able to store the information and ultimately to validate it and then to integrate it and ultimately to image and model the information. And of course, what we're really struggling with now and can use a lot of help from you is how we take the 30 or so individual programs we've developed and put them into an integrated architecture and make the engineering sufficiently robust that even biologists can do these complicated manipulations. The problem with federal funding is they don't ever give you the money for software engineering, and that's why we found partnering with appropriate industrial computer science companies has been very useful. So at ISB, we have systems that gather data. We put them through a pipeline of informatics, biological computational analyses, and then finally we output this information to simple and complex model systems and ultimately to understanding human biology. The fundamental hypothesis for a systems approach to medicine is the simple idea that disease arises as a consequence of one or more networks in the relevant organ that become disease perturbed. That, and it can be done, perturbations can come about through genetic modification or and/or environmental signals. The disease perturbed networks then express an altered envelope of information that changes dynamically across the course of disease progression. And you can use that altered envelope of information, number one, to understand mechanistically the nature of the disease, but number two, you can think about systems approaches to diagnosis, to therapy, and prevention. And in the course of my example, we'll talk about some of these possibilities. So I'm going to give you a little bit of biology, and I just want to make a few high-level points about how systems approach to disease is absolutely one of the key transformational events that's going to lead to the emergence of this P4 medicine. We studied an infectious disease in the brain, prion infections in mice, and we did it because we could initiate the infection and then we could look at how the information in the diseased organ, the brain, changed at multiple time points across the onset of the disease, and we could compare the diseased animals against their normal littermate counterparts to focus on just that differential envelope of information that is manifest and expressed. In the first set of experiments, we generated 50 million data sets. This was a couple of years ago. That was then an enormous amount of data. You'll see later it's a pretty modest amount of data. But the really key point is in these 50 million data points that changed in an interesting way across the onset of the disease, it turned out that most of those data points are noise. And the question is how you deal with signal to noise. So let me tell you, we dealt with it by looking at a whole series of different strains of mice that manipulated genes relevant to this disease process in very interesting ways, and by carrying out subtractive analyses of genetic mutant mice that did things that weren't core and central to the disease, we were able to take the 7,400 genes that changed in one or more of these 8 strains and reduce it to a core of about 300 genes that were key to this prion disease response. And then what we did was to map those 300 genes into 4 fundamental types of biological networks that had been defined by the histopathology of this disease: prion replication and accumulation, the activation of glial cells, macrophage-like cells, and then 2 aspects of neural degeneration. So let me show you dynamically how the information changes. So here's a network. Yellow indicates that the normal and diseased animals have RNA transcripts that are at the same level. This is at 6 weeks. The solid lines around subnetworks indicate specific biological functions within the context of that larger biological network. What you can see is at 10 weeks, things have begun to change. The red dots indicate elevation of information levels in those transcripts. And at 14 weeks, you can see even more massive changes. And 14 weeks here is 4 weeks before any clinical signs show up in this disease whatsoever. 2 weeks before the animals are exsanguinated, or 20 weeks, you can see massive changes that have occurred here. Hence, we have for each of these animals 10 different time points across each of these 4 different types of networks. And the general conclusions that we can draw are really quite striking. 2/3 of the 300 genes mapped into the 4 known networks, and those networks explain every single aspect of the pathophysiology of prion disease that we previously had known. The other 100 genes were, in a sense, the dark genes, the dark matter genes of prion disease. They encoded modules that participated in the disease process that heretofore had not been known. And the reason we identified them is because these analyses were global. We looked at everything and we inferred that anything that changed had to be a part of this disease process after these subtractive analyses. What is really clear, and I'll show that in a moment, is these 4 networks are sequentially activated with regard to disease perturbation. And what we'd like to do is look at the first network that is specific to the disease and be able with drugs to re-engineer it so we can abrogate the downstream process that leads to neuron destruction and ultimately to neuron death. And of course, the other thing this did is it started us thinking about a systems approach to diagnostics. And let me just say, blood diagnostics is going to be the framework of P4 medicine over the next 5 to 10 years, and I hope to give you a sense of why this is so important. So if we look at 3 panels from one of these networks, we can see at the 12-week panel, there are changes that occur in the network 8 weeks before any clinical signs show up, 6 weeks show up before any clinical signs show up. So the question is, would these transcripts encode proteins that are secreted into the blood, and could their altered levels reflect the disease process? And the answer to that is beautifully yes. But you need one more thing for a systems approach to disease. You need to be able to know where those changes of blood proteins are localized with regard to organ origin. And the approach that we took there was to develop genomic techniques. So for each of the 50 or so human and mouse organs, we were able to identify transcripts that were organ-specific, or at least relatively organ-specific. And then we used classic proteomics techniques to identify which of those organ-specific proteins showed up in the blood. And each of the set of, in several cases now, we have 25 to 50 protein fingerprints in the blood for the brain and the liver. Each of those proteins reflects, obviously, the operation of a particular cognate biological network. If it's normal, the level of that protein will be at one stage. If that network becomes disease-perturbed, then that protein level will change up or down. And the important point is different diseases perturb different combinations of networks. Hence, in these fingerprints, each will uniquely separate health from disease, and if a disease, it will give a unique fingerprint for each specific type of disease that's present in that organ. And indeed, what we were able to demonstrate with prion disease, which is really exciting, is a sequential activation of these 4 different networks that I showed you earlier. And we can identify blood proteins that are encoded by each of those 4 successive networks whose levels change in the blood at the time those networks become disease perturbed. So from the blood, we can follow the entire disease process, and this will absolutely transform how we think about doing drug target discovery in the future, as well as giving us the ability to do a whole series of things with disease diagnostics. One, we can do pre-symptomatic or very early diagnosis, absolutely critical for cancer. Number 2, we can stratify disease. If you take something like breast cancer, it's probably 4 or 5 different diseases. And what we want to be able to do from the blood is stratify in each woman which of the 4 or 5 types she has, and then carry out an impedance match against appropriate drugs that will be able to achieve very high levels of cure rates. And that's what we're in the process of doing now. As I showed you earlier with the prion disease, we can assess the stage of progression of the disease from the blood, and in the future, your treatments will change according to the stage of progression. And of course, we can use these diagnostics to follow therapy. And it's these initiatives that this company you heard earlier about, Integrated Diagnostics, was founded to create. We think it has a chance of being a fundamental platform company in the emergence of P4 medicine. So technologies change biology, and even more are going to change medicine in major ways. And let me talk just briefly about 3 technologies that we've been working on. Sequencing, microfluidic chips that can measure blood protein levels, and single-cell analyses. Together with Complete Genomics, we've recently carried out the total genomic sequence analysis of a Caucasian family of 4. The mother and father were normal. The 2 kids each had 2 genetic diseases, a craniofacial malformation and a cystic fibrosis like lung disease. And what we were able to demonstrate is the complete genome sequence of this allowed us actually to carry out, there we go. Allowed us to carry out an analysis using simple principles of Mendelian genetics and the inheritance of genes to, by more than 10-fold, reduce the signal-to-noise errors intrinsic in large-scale DNA sequencing. And this increased accuracy allowed us to, in one family, identify 4 gene candidates, 2 of which turned out to be the causative genes for each of these 2 diseases, and the other 2 were almost certainly really critical modifier genes. So family genome sequencing is the way all genome sequencing is going to be done in the future. It is the most efficient way by far to find disease genes. Microfluidic chips, we'd like to be able to measure these blood proteins, and together with Jim Heath at Caltech, we've developed a microfluidic approach that allows us to take 300 nanoliters of blood, a fraction of a droplet of blood, and make 35 measurements in less than 5 minutes with a high level of sensitivity and a dynamic range that's appropriate for the complexity of blood. We have these chips in clinical tests now at UCLA. We're looking at patients with melanoma and glioblastoma and their responses to different drugs. And of course, what's great about the chips is it's a simple pin prick, and you can do the analyses, so you can do very rapid kinetics. The challenge with these chips is to get the protein capture agents that are the locus for measurement, and we're developing new strategies that will, I think, totally within 5 years replace antibodies as protein capture agents of the future. And finally, single-cell analyses, I think, are utterly going to be transformational in biology and in medicine in interesting ways that we can talk about now. So let me run through 6 measurements that we're beginning to apply to patients, and in all cases, we've done the preliminary work to give you some idea about how medicine of the future really is going to become informational. So, number 1, I'd argue in 10 years, the genome sequence of an individual will be $300 or $400, and all of us will have those done in the context of our families, and that will be a base core part of your medical record from which you can begin to think about deriving strategies for lifelong health management, something we've never been able to think about doing before. Number 2, we're going to be able to do the entire genome sequence from one cell. And this will, for example, transform our understanding of cancer and what the cancer process is now. There's a big project called the Cancer Atlas Project that takes tumors and does DNA sequencing, averaging. There are many different types of cells. And I've argued for a long time that is basically a useless approach because it doesn't give you the quantized information you need to understand the key cellular subtypes that are fundamental constituents of the tumors. Within 2 years, I predict we can take 1,000 cells from one tumor. We can analyze complete transcriptomes, looking at 25,000 different RNA, messenger RNAs. And we can do those 1,000-cell analyses after appropriate barcoding in a single run of a DNA sequencer. So this will transform the level at which we can begin to do systems biology in biology and obviously in medicine. I predict in 5 to 8 years we'll have handheld devices that can prick your thumb, take the measurement of 2,500 proteins, and I would argue there'll be 50 proteins from each of your 50 major organ systems, and you'll do a wellness against disease assessment for each of these organs, and you'll do it twice a year. If the report comes back to you and your physician, you're fine, do it again in 6 months, that's one thing. Or it could come back, you'd better see your rheumatologist and see what's going on. But this is going to allow us to focus on the idea of wellness in a way we've never been able to do before. We'll be able to take 10,000 T cells and 10,000 B cells and assess your past immunologic history, your present immunologic state of readiness, and we'll be able to assess how well vaccines are actually working in you. And I think that can really transform how we deal with the immune system and so forth. And finally, I think most striking in many ways, from your blood, we'll be able to convert nucleated white cells into iPS cells, which are stem cells. So every individual will have stored away stem cells that then can be differentiated to whatever tissue is relevant for diseases of the individual or their family. And then we can interrogate in vitro these relevant tissues using detailed molecular imaging or higher level phenotypic measurements. So it's the systems approach to disease plus these emerging technologies that will in fact give us P4 medicine. So the predictive part I've already talked about. We'll have your genome sequences, and from that make predictions about future health history with appropriate information that assesses environmental impact on the digital genome. Number 2, we'll be able to do twice a year these 2,500 blood protein measurements and assess your wellness against disease progression and so forth. The personalized part of medicine arises because Each of you differs by 6 million nucleotides on average from your next door neighbor. And as a medical student at Hopkins, I never understood the idea that you could take the parameters of a large population of patients and you could create averages, and you could say if you fell outside these averages you were sick or you're healthy. The real fact is every individual will be their own control for whether they're healthy or whether they're diseased. The longitudinal information we'll acquire on each of you as individuals will be the real source point for establishing the baseline for wellness. Now, imagine this. As I said, in 10 years, we'll have billions of data points on each of you. So again, we'll be able to search wellness and disease space in ways we've never conceived. But even more important, we'll have hundreds of millions of patients where we have billions of data points. And it's this information, if we have access to it with the kind of tools you can provide, that will bootstrap for us the predictive, preventive medicine of the future. And what I fear is, will we have standardization rules? Will we have interoperability of software? Will we have accessibility? Can we deal with the ethical laws and so forth? These are all the things that we really have to worry about. Preventive medicine will have completely new strategies for developing drugs that will operate on small populations of patients and still make the drug companies money. We'll be able to use systems approaches to understand the immune system and effectively create vaccines. I mean, we've spent billions of dollars down a black hole on AIDS vaccines for the last 10 years, with the pharmaceutical companies basically doing exactly what Jenner did in 1796 in terms of immunization. So it's time to understand the immune system and from that figure out how to do vaccines, not to do the same thing over and over again unsuccessfully in the case of AIDS. And of course, the real transformation here is going to be the move from a focus in disease to a focus on wellness. That's really going to be a key part of the future. So participation, how do we educate patients both as to their opportunities and their responsibilities? How do we educate physicians that largely will have been trained prior to the explosive emergence of P4 medicine? How are we going to create a medical community that can be appropriately interconnected and understanding of these new opportunities? I would say those are all basically IT software kinds of problems. And let me give you a series of fundamental challenges. So one challenge is, how do we take basic information, translational information, and the core information of digital biological records today and integrate them together in a seamless fashion where we can migrate inwardly the procedures as they become standard process. We're not even thinking about how to do that, that whole endeavor. How are we going to create information that is going to give us access to pipelines and data and records and all the other kinds of things? How are we going to carry out the educational processes that we've already talked about that are going to be critical? And I would argue a lot of education is going to come down to things we can do with games and new ways of entertainment that could be, I think, very, very powerful. And of course, how do we put together all of these different systems? How do we deal with security issues, all the rest? So IT for healthcare, what worries me about the Obama administration is they're focusing the IT on medicine of the past. And frankly, that medicine of the past, even if you do a good job on it, we're going to throw that IT away in 5 years because it isn't going to be able to deal with the kind of P4 medicine that we're talking about out here. So there are many different interesting IT challenges. So 2 further points about the impact of P4 medicine on society. Over the next 10 years, P4 medicine will force every single sector of the healthcare industry to radically alter their business plans and to adapt entirely new strategies for dealing with informational medicine. And I think the really interesting question is, how many of these old organizations are going to be able to adapt, and how many will be replaced by agile new organizations? And in my experience, old organizations with pre-existing bureaucracy really have trouble changing. The second point is that we're going to see a digitalization of medicine that is unlike anything we've seen before. We'll be able, from one molecule, one cell, one organ, to infer disease-relevant kinds of information. And of course, the digitalization of medicine will have the same implication that the digitalization of IT and communications has had. I predict there will be a time in the future when there'll be a sharp breakpoint in the ever-escalating costs of healthcare, and it will go down, in fact, to the point where we can export P4 medicine to the developing world, and it will be the framework for global medicine. And indeed, what's exciting about that to me is, for the first time, we can think about the democratization of healthcare in an unparalleled manner and everything. So how are we going to go about accelerating the time at which this breakpoint occurs and a real revolution begins? We've thought about it in terms of the alignment of critical strategic partnerships that bring together very, very effective on a worldwide basis people who have skills and technologies and computational tools that can focus on central issues in P4 medicine, together with our ability to deal with the societal issues that emerge from P4 medicine. So, for example, last year we consummated an agreement with the state of Luxembourg that is going to bring in roughly $100 million to ISB over the next 5 years to focus on the two fundamental problems in P4 medicine. I'd be glad to tell anybody who's interested what those are. And we're going to have a collaboration with Ohio State University, which is a unique organization because its 55,000 employees are managed by the medical school itself. That is, they are the payer provider for the medical school. So together with Ohio State, we're going to set up 2 pilot projects, one in chronic medicine and one in wellness, that will use those patients to introduce all of these new types of technologies and approaches to understanding disease and wellness that we've talked about here. And I think pilot projects with killer applications are the way you change things. Even in the midst of resistance from regulation, resistance from insurance companies and the all, success is pretty hard to deny. So let me close then with just a couple of general comments about computational challenges in biology at a high level. So one issue is data space is infinite. So if we want to search relevant data space for biology or disease, your searches have to be incredibly carefully formulated. So there has to be a marriage of deep biology with deep computation, mathematical, and measurement kinds of tools. The global measurements of all these different types of data are really critical. How do we put together pipelines? How do we do validation? How do we make the measurements that will give us access to the true dimensionality of wellness space and data space? And the enormous dimensionality of information, obviously, your genomes, we have to be able to reduce that enormous dimensionality to simple hypotheses about health and disease, and we do that statistically, but we also do it with really clever biology, quite obviously. And how do we integrate the different types of information together, ultimately to create the models that are both predictive and, from the point of view of medicine, are actually actionable? And the final point I'd make is that there are really interesting considerations about the granularity of information you need. And what's comforting about medicine is it's far less granular than a detailed understanding of mechanisms. Rather, all we need is the ability to make the pattern correlations and so forth. So in closing, I would just say that this medicine of the future will be information-based, and computer science, and in particular high-throughput computing are going to play central and fascinating roles in letting us deal effectively with this information and with the ability to integrate and ultimately make hypotheses about this information. I think there are enormous challenges. I think there are enormous opportunities, and I think in this area the world will really change over the next 5 years or so. Thank you very much. Time to go on with questions.
Okay. Thank you very much, Lee. I think we have just a little amount of time for one or two questions. There's a mic here and a mic there. Oh, there's three. So if you can quickly make it to the mic, I can squeeze one or two in. Yes, number two.
Just a question on the prion study. Were you able to separate out the immunological response from the disease response?
So the answer is we were. Glial activation is basically the activation of innate immunity and complement and things like that, and we can sort that out beautifully. But I would say that immune response is a fundamental part of the disease process. It's not something that's separate and apart, but it's integrated into the immune response, and its activation is almost certainly part of what ultimately leads to neural degeneration and apoptosis or neural death and everything. But the beauty of this is you could beautifully see the sequential recruitment of the various elements of this disease process across time. And the reason I emphasize the importance of being able to see that recruitment in the blood is we're now in the process of studying human neurologic degenerative diseases, and we aren't going to have access to the brains and the information in the brains, but we will have access to the information in the blood.
Thank you.
And another question on mic 3, and I think that's probably all we're going to be able to have.
Dr. Hood, thanks for a really nice talk. You mentioned that one of the big problems with new high-throughput genome sequencing is that the reads are very noisy, and that, at least for individuals, you can get a lot of benefit by integrating information from a family tree. I'm wondering if you think that there's a similar approach you can use to solve the noise problem when you're doing reads on cancers, as you mentioned, because the inheritance rules are obviously going to be highly distorted there with all the genomic rearrangements.
Well, you know, I think you won't be able to apply those principles to cancer because, you know, the essence of cancer cells is their mutational rate is increased by 400-fold, and to a first approximation, it's random. So I think what's really going to transform our understanding of cancer is the ability to look in a particular tumor at 1,000 different cancer cells and interrogate their genomic, their proteomic, and even transcriptome information in enormous detail so that we can understand the nature of the quantized cellular states that exist within the tumor and begin to understand how they interact with one another and enhance the whole process. But superimposed on the events that lead to cancer death is an enormous random mutational process, and with single-cell analyses, we can sort out what's relevant and what isn't relevant in that regard. You can't do it by taking 100 million cells and scrunching them all together and then averaging across populations. You miss the essence of the unique biological dimensionality of the disease.
Thank you very much for your questions. I'll have to stop right there. Please join me in thanking Dr. Hood.